Welcome back, everyone, for another deep dive. This time we're exploring a bacterium, and I think if you're a medical microbiologist, you're probably pretty familiar with this one. It's Alloiococcus otitidis.
Oh, yeah.
And to help us with this deep dive, we've got a couple of great resources. We've got Microbe Wiki, Microbe Canvas, and an interesting article from the Journal of Clinical Microbiology.
Awesome.
Yeah, so we're going to become otitis experts today.
Sounds good.
And then we're going to put all that knowledge to the test with some exam-style questions.
I'm ready.
Okay, great. So why don't we start with the basics? What exactly is otitis, and, why should we care?
Well, Otitis is a gram-positive bacterium, and it belongs to the family Carnobacteriaceae. And it's a really clinically significant bacterium, especially in pediatric cases.
Okay, so that's where I come in then as a medical microbiologist?
Exactly.
We see a lot of ear infections in kids, so is that what we're talking about here?
Yeah, you got it. It's heavily linked to otitis media, and specifically otitis media with effusion, or OME. So that fluid buildup in the middle ear.
Right, and OME is so common, especially in younger children.
Yeah. And
it can really impact their development if it's not addressed properly. I got to admit, I'm kind of surprised to hear that Otitis is such a key player here.
Yeah.
What makes it so unique in causing OME?
Well, one of the things that's really interesting about it is that it can persist even after antibiotic treatment.
Oh, really?
Yeah. So there's an article, the Journal of Clinical Microbiology article, actually highlights a study where they found otitis in children with OME, even after they had a course of antibiotics.
Wow, that's really interesting. And I think it challenges a lot of our assumptions about how effective antibiotics are.
Yeah.
And it raises some interesting questions about treatment strategies, because we know that if OME isn't diagnosed and treated quickly, it can lead to long-term consequences, especially for language development. So how does this persistence, this ability for it to stick around, how does that factor into the clinical presentation of OME? What kind of challenges does that create for us in a clinical setting?
Well, it definitely complicates things,
yeah,
because it means that we can't always rely on just, giving a standard course of antibiotics and expecting a resolution.
Right.
And so we have to be more vigilant about monitoring, these cases,
yeah,
considering that it might recur and that we might have to, look at alternative treatment approaches.
So as a medical microbiologist, what should I be looking out for in terms of trying to identify Otitis in cultures?
Well, one of the things you need to remember about Otitis is that it can be a little bit tricky to culture.
Okay.
It grows really slowly.
Okay.
And it often takes, 4 or 5 days for colonies to appear.
4 or 5 days?
Yeah.
That's a long time to wait.
It is.
Especially when you've got a concerned parent and, a potentially uncomfortable child.
Yeah.
So are there any characteristics or anything that can help us, distinguish Otitis from other bacteria that might be present in your cultures?
Yeah, absolutely. So under the microscope, Otitis appears as gram-positive ovoid culture that are typically arranged in pairs or clusters.
Okay.
It's an obligate aerobe, so it needs oxygen to grow.
Right.
And one thing that's helpful to know is that it doesn't grow on brain-heart infusion agar with 5% sheep blood.
Oh, interesting.
So BB.
So that's like a red flag, right? If we don't see any growth on BB?
Exactly.
Could point towards otitis.
It could help you narrow it down.
Okay.
And another thing to remember is that, we can't only rely on culturing to identify it.
Right.
If you remember that Journal of Clinical Microbiology study, they found that PCR testing was a lot more effective.
Oh, right. Yeah.
Especially in cases where antibiotics had already been used.
Because it can detect the bacterial DNA, even if it's not, actively growing in the culture.
Exactly.
So PCR might be a better tool to use in those cases where, Otitis is kind of hiding out.
Exactly. It can help us get a more accurate picture.
Yeah. Okay, great. So that's good to know. I'm also curious about just some of its other characteristics. We've talked about the gram stain, we've talked about the oxygen requirements.
Yeah.
But what else makes this bacterium tick? What are some of its other defining features?
Well, from what we know, it typically forms small alpha hemolytic colonies on blood agar and brain-heart infusion agar.
Okay, alpha hemolytic. Now, remind me, what does that mean again?
So alpha hemolysis means that it only partially breaks down the red blood cells in the agar.
Okay.
So it creates this greenish zone around the colonies.
It's like a little halo around the colonies.
Yeah, exactly.
Okay, so we've got the gram stain, we've got the oxygen preference, we've got the slow growth, the alpha hemolysis. What else do we keep in mind?
It's also catalase positive and oxidase negative.
Okay.
And those biochemical tests can help us, further classify and identify it.
Great. So I think we've got a pretty good handle now on Aotititis, from its role in OME to its, culture characteristics and biochemical reactions.
I think so.
Are you ready to put our knowledge for the test?
Yes, bring on the exam questions.
All right, let's do it. All right, so first question, what is the primary clinical manifestation associated with Alloiococcus otitidis infection?
Well, based on what we've talked about, the primary one is otitis media with effusion, or OME.
Right.
That fluid buildup in the middle ear.
Okay, good. Next question. What is the gram stain morphology of Alloiococcus otitidis?
So on a gram stain, we'd see gram-positive ovoid cocci arranged in pairs and cluster.
Okay, excellent. Question number 3. Why is culturing Alloiococcus otitidis challenging?
Oh, this is a good one for medical microbiologists, right?
Right.
Well, one of the biggest challenges is it's really slow growing.
Yeah.
It can take, up to 5 days for those colonies to show up.
Right.
And that, can make it difficult when you're trying to make treatment decisions.
Yeah, for sure.
It also has those, specific media requirements that we talked about. So that can make it a little bit finicky.
It definitely tests our patience.
It does.
Okay, question number 4. What key antibiotic susceptibility pattern distinguishes Alloiococcus otitidis from other common middle ear pathogens?
All right, so A. otitidis is susceptible to vancomycin.
Okay.
But it's resistant to colistin.
Interesting.
And that's something that's really important, when we're trying to identify it and figure out what to treat it with.
Yeah, for sure. For sure. All right, last question. Question number 5. Why might PCR be a preferred method for detecting Alloiococcus otitidis in clinical samples?
Well, thinking back to that Journal of Clinical Microbiology article that we talked about, they actually showed that PCR was more effective at detecting it, especially in cases where antibiotics had already been used.
Okay.
And that's because PCR can detect that bacterial DNA,
Right.
even if it's not actively growing in the culture.
Right. It's like a little secret weapon.
Exactly.
For those tricky cases.
Exactly. Gives us a better advantage.
Yeah. Okay, great. Well, you aged all the questions.
Woo!
I'm impressed.
Thanks.
It's really amazing, how such a tiny organism can have such big implications for, how we manage patients.
It is. It really highlights why we can't underestimate these bugs.
Absolutely. And why we need to, constantly be learning about them.
For sure.
So, speaking of patient care, I keep thinking back to that study from the Journal of Clinical Microbiology.
Yeah.
The fact that A. otitidis can persist even after antibiotic treatment,
Yeah.
it kind of changes the game a little bit, doesn't it?
It does. It makes us wonder, are we using the right antibiotics?
Right.
Are there other approaches that we should be considering?
Yeah, as a medical microbiologist, those are the kind of questions I ask myself all the time, right?
Yeah.
We want what's best for our patients, and especially, those little kids
Yeah.
who are really vulnerable to the effects of OME. So what are your thoughts? Where do we go from here?
Well, I think we have to stay curious and keep learning.
Yeah.
The research on otitis is ongoing.
Right.
And there's still a lot that we don't know. So
Yeah.
I think we need to, delve deeper into how it persists, how it interacts with other microbes in the middle ear.
Okay.
And, potentially investigate some new therapeutic targets.
It's almost like we're scientific detectives, right?
It is.
We're trying to piece together these clues and solve the mystery
I like that analogy.
of otitidis and its role in OME.
Yeah, and the more that we understand,
Yeah.
the better equipped we'll be to find, more effective ways
Right.
to treat these infections.
Absolutely. Yeah. So, maybe we'll find some new antibiotic combinations.
Yeah.
Or maybe we'll look at things like probiotics and how that can, restore that balance in the middle ear.
Right.
Or maybe we'll even be able to find ways to kind of modulate the host's immune response.
Yeah, the possibilities are exciting.
It is exciting to think about, this deep dive has really been eye-opening for me.
Yeah.
It just makes you realize how complex Aotititis is.
Yeah.
And how much more research is needed.
Absolutely.
So I'm definitely walking away with a renewed sense of curiosity.
Good.
And definitely a drive to learn more.
That's what it's all about. Never stop questioning.
Well said.
Never stop exploring.
Well, it's been a pleasure dissecting Aotititis with you today. Thank you so much for sharing all of your knowledge and insights.
It's been my pleasure.
And to you, our listener, thank you for joining us on this little microbial adventure.
Thanks for listening.
We hope you've learned a lot about Aotititis and how it impacts patient care.
Yeah, keep those microscopes sharp.
Stay curious and keep on learning. It's amazing how much we've learned about Aotititis in such a short time.
I know.
It's really fascinating. We went from the basic description to really understanding its clinical significance, the challenges that it presents.
Yeah, and I think it just shows us that we should never underestimate, Even the simplest microbes,
Right.
there's always something new to discover.
Yeah, and that knowledge, it directly impacts how we take care of our patients.
Absolutely.
And speaking of patient care, I keep thinking about that Journal of Clinical Microbiology study.
Yeah.
And that otitis can persist even after antibiotic treatment.
Mm-hmm.
I mean, that's a game changer.
It really is. It kind of raises the question of, are we doing the right thing?
Yeah.
Are we using the right antibiotics? Are there other things that we should be considering?
And as a medical microbiologist, these are the kind of questions that I think about all the time.
Yeah.
We want the best for our patients, especially these little kids
Right.
who are so vulnerable when it comes to OME. So where do we go from here? What are your thoughts on this?
Well, I think, first of all, we just have to stay curious
Okay.
and keep learning. The research is ongoing, and there's still so much that we don't know about this bacterium.
Right.
So I think, we need to delve a little bit deeper into those mechanisms, the mechanisms of persistence. How does it interact with other microbes in the middle ear?
Right.
And, maybe look at some new therapeutic targets.
Yeah, it's like we're scientific detectives, right?
Yeah, it is kind of like that.
Trying to, put all the pieces together, solve the mystery of a otitis and its role in OME.
Yeah, exactly. And the more we learn, the
Yeah.
better equipped we'll be to, manage these infections properly.
Yeah, absolutely. So, maybe we'll discover new antibiotic combinations, or maybe we'll look at things like probiotics, and how that can restore the balance in the middle ear. Or maybe we'll even find ways to modulate the host immune response.
Yeah, the possibilities are endless. It's very exciting.
It is exciting. Yeah. This deep dive for me has really been eye-opening.
Good.
It's made me realize just how complex otitis is.
Yeah.
And how much, more research is needed.
For sure.
I'm walking away with a renewed sense of curiosity
That's great.
and definitely a drive to learn more.
And that's what it's all about, right? Never stop learning, never stop questioning.
Well said. Well, it has been an absolute pleasure, kind of dissecting otitis with you today. Thank you so much for sharing your expertise, your insights.
Oh, it's been my pleasure.
And to all of our listeners out there, thanks for joining us on this little microbial adventure.
Yeah, thanks for listening, everyone.
We hope you've learned a lot about otitis and, how it can impact patient care.
Keep those microscopes sharp.
Stay curious and keep on learning.